Dr. Swati Sharma from DRC-DU. Today we would be discussing the media preparation. First of all, you should know what is a media. Media is a medium in which you can grow your microorganism of the interest. In microbiology, you want to culture your microbiome of your interest to study it. So there are 5 basic techniques by which you can study a microorganism. The first technique is to culture them, and there are 5 ways or 5 different steps for culturing. First is inoculate. You have to select the microorganism of your interest and then inoculate in a culture media. The second step is to incubate. Each microorganism has a specific temperature and pH on which it can grow and thrive. So you will incubate your culture at that pH and temperature for a prolonged period of time, such as 12 to 16 hours. So that is called incubation. The third step is isolate. Once after incubation you have a good bacterial culture, you will isolate your bacteria or your microbial colony of interest. Then you will inspect whether what you have isolated is pure or not, it is free from contamination or not, and then you will identify which culture you was using. Is it the same or it has got certain mutation? So these are the 5 I's of the culture media preparation. So if you want to prepare the media, you have to know the requirement. The media requirement depends upon the type of the microorganism which is being used in the fermentation process, but the basic requirement remain the same for most of them, which includes first a source of energy, because all of the cells, all the microorganism, they need energy to replicate, to divide. Second is water and the carbon source, because carbon is the backbone of not only their genomic DNA or RNA but also the macromolecules. Third is the nitrogen source, and then vitamins and the minerals. So designing the media for the small scale lab purpose is very easy, but the media for the industrial purposes are difficult to prepare because you have to do it for the large scale and in the large volume. So everything needs to be standardized. An ideal culture medium has the following features. A. It allows the high yield of the desired product at the fast rate. B. It allows the low yield of the undesired products or the toxin. C. It can be sterilized easily. D. The yield is consistent. The products are same. So it have minimum batch to batch variation. E. It's cheap and readily available. F. It's compatible with the fermentation process or the upscale process. G. It does not pose any environmental problem or the risk before, during or after the fermentation process. So the culture medium will affect the design of the fermenter. For example, hydrocarbons in the media require high oxygen content. So an air-lift fermenter should be used. Natural media ingredients are cheap, but they have high batch to batch variation. On the other hand, pure ingredients, also called the defined media or the formulated media, have very little batch to batch variation, but they are really expensive. So what can be the carbon and energy source, nitrogen source, nutrient source? These would be provided in the media so that you will get your product of interest, which is produced by the microorganism, along with the byproducts such as carbon dioxide, water, heat and biomass. The components of the medias are designed on the above equation using the minimum component required to produce the maximum product yield. So this is the equation. You can note down this equation: carbon plus energy source plus nitrogen source plus nutrients. They will give rise to product carbon dioxide, water, heat and biomass. So important components of the medias are discussed as follows. First, carbon source. The product formation is directly dependent on the rate at which the carbon source is metabolized. Also, the main product of the fermentation determine the type of the carbon source to be used. Carbon sources includes carbohydrates, oils, fats and hydrocarbons. Let's move with the carbohydrates. These are the most commonly used carbon source in the fermentation process. Starch is easily available carbohydrate obtained from the maize, cereals and potatoes. It is widely used in alcohol fermentation. Grains like maize are used directly in the form of ground powder as carbohydrate. Malt and beer are made from barley grains that contain high concentration of different carbohydrates such as starch, sucrose, cellulose and other sugars. Sucrose is obtained from the sugar cane and molasses. Molasses is one of the cheapest source of the carbohydrate. It contains high sugar concentration and other components like nitrogenous substances and vitamins, and it is used in alcohol, single cell protein amino acid and organic acid fermentations. Extractions and purification of the product is very expensive. Sulfate waste liquid is the byproduct of the paper industry. It contains carbohydrates and it is used in yeast cultivation. Whey is the byproduct of the dairy industry. It is used in alcohol, SCP, gum, vitamins and lactic acid fermentation. The other source of the carbohydrates can be oils and the fats. Vegetable oils are used as a carbon source. Oils provide more energy per weight compared to the sugar. They are also anti-foaming properties, but they are generally used as additives rather than the sole carbon source. Examples are olive oil, cottonseed oil, soybean oil, linseed oil, etc. Hydrocarbon can also serve as the carbon source. C12 to C18 alkanes can be used as carbon source. They are cheap. They have more carbon energy per content per weight than the sugars. They can be used in the organic acids, amino acids, antibiotics, enzymes and protein fermentation. Nitrogen sources. So, most commonly used nitrogen sources are ammonia, ammonium salts and urea, and they are used in the fermentation process. Ammonia also serve as the purpose of pH control. The other substances which are used as nitrogen sources are corn strip liquor, soya meal, peanut meal, cottonseed meal, amino acids and proteins. The third, minerals. Calcium, chlorine, magnesium, phosphorus, potassium and sulfur are the essential minerals for all the media because they are required by all the microbes. The other minerals like copper, cobalt, iron, manganese, molybdenum and zinc are the trace elements which are needed in minute quantity, and they can be present as impurity and other component. The specific concentration of these elements depend upon the microorganism which is being cultured or being used. The other thing that we need is growth factors. Vitamins, amino acids and fatty acids are used as the growth factors in the fermentation process to complement the cell component of the microorganism. Then we have chelating agents. The chelating agents prevent the formation of the insoluble metal precipitate. They form the complexes with the metal ions present in the medium and can be utilized by the microorganism. Chelating agents are not required in the large scale fermentation process since some of the other ingredients like yeast extract will perform the function of forming the complexes with the metal ions. One of the example of the chelating agent is EDTA. EDTA is a versatile chelating agent and it forms 6 bonds with the metal ions. It is frequently used in soaps and detergents because it forms a complex with calcium ions and magnesium ions. These ions are in the hard water and interfere with the cleaning agent of the soap and detergent. Other chelating agents are citric acid and pyrophosphates. Buffers. Buffers are used to maintain the pH of the medium as the microbial growth is affected by the pH changes. Optimum pH for the most of the microorganism is the 7. Commonly used buffers are the calcium carbonate, ammonia and sodium hydroxide. Antifoaming agents. The microbial produces a large amount of the foam in the fermentation vessel. This is due to the microbial proteins or the other components of the media. Foaming causes the removal of the cells from the media and their autolysis, thus releasing more microbial foam producing proteins and aggravating the problem. Foam will reduce the working volume in the fermentation vessel. decrease the rate of heat transfer and deposit cell on the top of the fermenter. The air filter exists and then become the wet, allowing the growth of the contaminating microorganism. Antiforming agents are called surfactant because they reduce the surface tension in the foam and destabilize the foam producing proteins. The commonly used antiforming agents are stearyl alcohol, cottonseed oil, linseed oil, olive oil, castor oil, soybean oil, cod liver oil, silicones and sulfonates. The air is required for aeration and is supplied to the fermenter by the means of the pumps or compressor. It is sterilized by the passing through the filters before being introduced. The amount of air required and the extent of the purity depend upon the fermentation process being carried out. Steam is usually used to sterilize fermenters and other equipments. To control temperature, continuous dry steam is required for the fermentation process. Care should be taken to prevent the condensation. The factors that influence the microbial growth are temperature, water, pH, nutrient, etc. Let's first discuss the temperature. Most microorganisms that are mesophiles require temperature 25 to 40 degrees Celsius for their optimum growth. As the temperature will increase or decrease, the growth rate will be affected. The growth rate will decrease. The thermophile are the class of the microbes that require high temperature, which is over 50 degrees Celsius. They cannot survive at the low temperature. The psychrophiles require very low temperature, which is minus 15 to 20 degrees Celsius for their survival and the growth. They love low temperature. They cannot grow in high temperature. The second requirement for the optimum growth is water. The microorganisms require optimum amount of the water or humidity to maintain their metabolism and produce required products. The third requirement is of optimum pH. The pH for the most of the bacteria is 6.5 to 7.5, but for yeast it's 4 to 5, for the molds which are fungi it's 4 to 7. The pH of the medium should be always maintained at the optimum level for the microorganism that is being employed to grow to ensure the better product yield. So, the pH of the media will depend upon the choice of the microorganism which you want to grow or culture. The other optimum requirement for growth is the nutrients. The microbes require optimum concentration of the nutrients like nitrogen, vitamins, minerals for their maximum growth rate. This will be different for each of the different type of the microbe. The optimum concentration of the nitrogen is ranging from 0.1 to 1 mg per liter. So, as you can see this chart, there are different kind of the media depending upon what you are using in the media. So, if your media is derived from the livings or the non-living, the media which is derived from the non-living can be considered as the natural media or the synthetic media. The media from which is derived from the living, it's the tissue culture media. So, let's classify media first basis upon their physical property or their consistency. So, based upon their consistency, the media can be of different types. Based upon its physical state, it can be either a liquid media or solid media. Based upon its chemical composition, it can be simple media or complex media. Based upon the functional type, it can be the nutrition media, storage media or selective media. Let's move on to first type, that is based upon the consistency. So, solid media are the media which are solid. Advantages of using solid media is that microbes may be identified by studying the colony. The mixed bacteria can be separated and pure colonies can be obtained. Solid media is used for the isolation of the microbe for the pure culture. Agar is the most commonly used reagent which is used for preparing the solid media. Agar is a polysaccharide which is extracted from the seaweed. It's an ideal solidifying agent because it is bacteriologically inert, that is no influence on the bacterial growth. It remains solid at 35 degrees Celsius. It is transparent. And the other example is the liquid media. It is used for the profuse growth when you need the large number of the cells. Example, blood culture in the liquid media. Mixed organism cannot be separated. So, this is the disadvantage of the liquid media. Based on the chemical composition media can be the routine lab media or the synthetic media. These are the synthetic media are usually chemically defined media which are prepared from the pure chemical substances. They are used in the research work. The routine lab media, they are not well defined. They are classified into 6 types. First, basal media, second, enriched media, third, selective media, fourth, indicator media, fifth, transport media, sixth, storage media. Basal media are those that may be used for the growth of the bacteria that do not exceed the enrichment of the media. Example, nutrient broth, nutrient agar and peptone water. Staphylococcus and enterobacteria grow in these media. Enriched media. They are the media which are usually used for enriching growth. They are enriched by adding blood, serum or egg. Example of them are blood agar and Lowenstein-Jenen media. Streptococci grow in the blood agar media. Selective media. These media are they favor the growth of a particular bacteria by inhibiting the growth of the undesired bacteria and allow the growth of the desired bacteria. Example, McConkey acar, telluride media. The telluride inhibits the growth of the most of the throat organism except diphtheria bacilli. Antibiotic may be added to the medium of the inhibition. Indicator or the differential media. Differential media are widely used for the differentiation of the closely related microorganism or the group of the organism. Because of the presence of certain dyes or chemicals in the media, the organism will produce certain characteristic changes or the growth patterns that are used for the identification or the differentiation of the microorganism, such as blood neutral red. Example is blood agar media, McConkey agar are the indicator media. Transfer media are used when specimen cannot be cultured soon after the collection. Example, Caribbear media, Ames medium, Straut's medium. Storage media. Media are used for the storing bacteria for a long period of time. Example, Exaline medium, chalk cooked meat broth. So these are the different type of the medias and their use. These are again different types and these are the nutrients and the ingredients. You can make the list of the nutrients and the ingredients. So these are the commercially available media that you can buy from various companies, and this is how, if your microbial culture is contaminated from any fungal infection, looks like. You can see the fungal growth in all of them. So if the sterilization techniques are not followed properly, it will result in the formation of the contamination, and this is how the contamination will look like. This is the composition of the various medias. These are the commercial available media, and the plate on the right show you the bacterial culture. This is again when the culture has been spoiled because of the contamination, when the sterilization techniques are not fully followed. You can see in the top there is the fungal contamination as it is spread, and in the top and in the bottom there is the formation of the callus or the crude bacterial growth. So this is the bacterial contamination. All these kind of contamination will ruin your experiments, and they can be avoided by following the proper disinfection techniques. So in case of the microbiology, you have to use both the combination of the physical and chemical disinfection techniques. So physiochemical techniques. This is how the fungal growth will look in the slides. So now let's discuss the growth media depending upon the functional type. Earlier we discussed the functional media depending upon the consistency. Now we are discussing based upon the function. So there are 3 different types: enriched media, selective media and differential media. The common fungal culture that are used are birdseed agar, and these are the essential ingredients, and the intended use is to use the selective and differential isolation of the Cryptococcus forms from the clinical specimens. The other is the brain heart infusion agar. The intended use is the growth of the pathogenic fungi such as Histoplasma capsitum and other dermatitis causing fungi. The other commonly used fungal media is the Candida media, and the intended use is the selective and differential chromogenic media for the isolation and identification of the various species of the Candida. Then common agar and spes agar dox media is also used. First is used for the identification of the albicanes by the microscopic morphology, and the second one, Cispac dox agar, is used for the identification of the Aspergillus and Penicillium species. It can also be used for the cladospore production by Candida albicans. Thank you. We'll end today's topic here, and here we finish our sterilization techniques and the cell culture media preparation and different type of the media.